Hello class. I hope everybody's doing good and you and your family are safe. And we are just possibly a couple of weeks away from finals. So we are also finishing the syllabus. And in next class, I will review the syllabus, how much we have covered. And so we have pretty much covered many things. And whatever is remaining will be covered in coming lectures. So today's class is on transgenic animals and medical biotechnology, as well as plant biotechnology. So I'd like to start with plant biotechnology first. So plants are unique living organisms in many ways, and they're very special because they can prepare their own food, and that food can be utilized by the secondary consumers. And not only that they provide food and nutrition, but they play a very important role in regulating the environmental gases. So plants are unique in many ways, and plants have become very important because of its value as food item, mainly, and the population is increasing, and there is scarcity of food. There could be so because most of the agricultural lands in many countries are being converted into several other things. So, but there's an alternative also, that's plant biotechnology, to increase the production. So if the population is increasing, the demand for food is directly proportional. So to keep up the demand, the only way is to use technology in plants. And the goal of the technology is first to produce, the second is to reduce the cost. But you can see that the cost is not really reducing. 90% of the plants or plant food or vegetables that we consume are most likely GMOs, and but the prices are not declining. On the other hand, if you compare to organic plants or organic vegetables, they are expensive as compared to the genetically modified organisms or plants. Second thing is that we always think or thought that GMOs, especially plants, are unhealthy. But so far, I haven't seen any concrete report. If you know, then kindly share with me. And so GMOs are it looks like they're safe. But yes, there are controversies and debate. Also, we should understand the biology of plants, because the plant is an open environment, and they can receive pollen grains from any other source. And so what I want to share with you, that plants have unique capability to undergo genetic recombination, because environment, birds, butterflies, wind, they all can carry the gametes, the pollen grain, to respective flower and can cause germination. So plants are undergoing recombination through various ways. And it's very interesting. I read somewhere, and I hope that information is correct, that broccoli is a genetically modified plant, right? So broccoli, and you know that broccoli is very, very good with regards to its nutritional value. So it's an interesting debate, and that's what science is about, because science exposes us to new things, and people think whether it is right or wrong, and that's how it should be. So plants are unique that they are totipotent. What do I mean by totipotent? That given the right condition, one can take plant tissue and can develop into an independent plant. And what are the three major reasons for developing a transgenic plant? Is to improve its agriculture value, nutritional value, horticulture value, or if it is an ornamental plant. Actually, plants are the first chemist or biochemist on this planet Earth. And so transgenic plant act as a living bioreactors for inexpensive production of economic imported proteins or metabolite. For example, many people are debating that whether intravenous vaccination of or intramuscular vaccination of COVID-19 effective. Maybe it could be given through nasal, or it could be edible vaccine, such that it could go directly into our system, into our internal body. So what can be helpful is to develop edible vaccines for not only for COVID-19, but for many other diseases, because it will be difficult to immunize children, small kids, to inject vaccines because it hurts. Right? It is interventional. So it makes logic to genetically alter plants to benefit human health and see if there is a cure element. Already there are a lot of medicinal plants which are of preventive value, and people are analyzing the therapeutic value of these medicinal plants. So why to genetically modify plants? Number one, to have insecticidal activity, protection from viral infections, because the way there are pandemics in human, there are pandemics in plants also, and it just wipes out all the crop. And many farmers, for many farmers or agriculturists, farming becomes a prime major profession. So also tolerant to extreme climatic conditions, to increase the nutritional value, edible vaccines to reduce the cost. So the one way that I have mentioned this briefly about TI plasmid, that there's a bacterium called as Agrobacterium tumefaciens, right? And this Agrobacterium tumefaciens, they have a special plasmid called as tDNA, and this plasmid is 10 to 30 kb, and the disease is caused by a virulence gene, VIR. And these Agrobacterium tumefaciens, they can cause tumor right at the junction of stem and root, and that can impede the growth of the plant. But scientists have utilized these agents to develop into tools, biotechnology tools. So they have utilized the T plasmid from the Agrobacterium tumefaciens to incorporate genes. And they have taken 2 steps, because all the cloning has to be done in E. coli. So this plasmid, which is without the VIR gene that causes tumor formation, has to be cloned in E. coli. Why E. coli? Because the gene, the foreign gene that would increase the value of plants, can only be cloned easily in E. coli. So it's a dual vector system where, number one, the TI plasmid or tDNA has to be manipulated to remove the pathogenic genes that causes tumor, and then replaced by some genes that would facilitate the propagation of this plasmid in the bacteria, so that the foreign gene could be inserted. So for molecular biology manipulation, E. coli, and for transformation into plants, Agrobacterium tumefaciens. And as I mentioned to you in the beginning, that really transgenic plants are at the peak of debate, and it is possible that these plants, these robustly designed transgenic plants, can cause a problem to let's say butterfly or insects. They can disrupt their life cycle. Right? It is possible that one layer of these small butterflies or insects could be totally wiped out from the planet. So in addition to genetically modified plants, The other approach could be the plant tissue culture, to take out the plant tissue and to grow in the lab condition and to produce plant and then take it to the field. And one of those techniques is micropropagation. It is a practice of rapidly multiplying stock plant material to produce large number of progeny plants during modern plant tissue culture. One of the challenges with micropropagation is that transitioning plants from lab to environment could be challenging, and there could be loss of, because there are experimental plants that are growing in artificial conditions, controlled condition, and when they are put into environment, they may not produce the same results as in closed environment. So micropropagation has various advantages. You can maintain the clones, meaning that if you're growing this plant internally, then you're avoiding genetic recombination. So you can have one breed of plant with equal nutritional value. That's the advantage of micropropagation. It's expensive because we provide artificial conditions. And so this was about plant biotechnology. Now I'm going into transgenic animals. It's very important that the most important tool that we need for creating transgenic plant or animal is a system that would help to carry the gene of interest into that organism. So let's say if we want to put GFP gene into bacteria, or we want to put insulin gene into bacteria. So one of the major vector would be plasmid that can carry the gene for insulin or GFP. So plasmids are extra chromosomal circular particles, or DNA, extra chromosomal circular double-stranded. These have antibiotic resistance marker. They have origin of replication so that they can replicate independent of the main DNA. So these plasmid, they have restriction sites. So this plasmid can be cut and the gene of interest can be inserted. So this was about bacteria. Similarly, we have plasmids for transfecting or transforming the mammalian cells or human cells. But the major difference between the bacterial plasmid and the mammalian cell plasmid is the promoter. The promoter for the eukaryotic or human system is cytomegalovirus. For bacterial, for bacterial transformation, we need bacterial plasmid with bacterial promoter. But for human cells or mammalian cells, we need plasmid with viral promoter, because viruses can express themselves inside the human cells. A human cell may not accept bacterial promoter to start the process of transcription. So these plasmid, they use viruses. In this situation, it is CMV, cytomegalovirus. This is the example of DNA, which has virulence region that has to be removed to be replaced by, let's say, auxin, cytokinins. These are plant hormones that can accelerate the growth of the plants, right? Also, then the vectors become really sophisticated when we go into making transgenic animals. And this has to be very carefully designed. It has to take care of the introns, because eukaryotic genes, they have introns, then there is elongation factor, then there are various regulatory regions, right? One of the most popular vectors that is used for transformation is lentivirus. And in this situation, these lentivirus vectors could be injected into the embryo and put back into mice. So embryo could be harvested. The gene of interest can be inserted into the retrovirus, and this retrovirus could be transfected or transduced into this embryo, and this embryo could be again put it into the mother's body. So what is the composition of lentivirus? A lentiviral vector is composed of two regions, LTR at 5' and 3'. This is called as a long terminal repeat, and they have an appropriate promoter, because promoter will facilitate transcription and translation. The transgene that has to be inserted is placed next to the promoter. And then this is the lentivirus. You know, lentivirus, they have to be packaged into viral particles. These viral particles then can enter into the cells. So these are psi, packaging signal, when PPT, polypropylene, tracked, so on and so forth. The other way it could be injected is to take out the fertilized egg, and then into the male nucleus, the transgene could be micro-injected. So micro-injection is one of the ways by which a foreign vector containing gene of interest could be injected into the nucleus. The third way could be to make into stem cells. If you remember, stem cells are present in blastocyst as inner mass cells. So these cells could be taken out, extracted, and then modified genetically, and then put back into the mother's body. Now these approach is particularly important for diseases which are very difficult to treat, as well as to understand its pathogenesis, especially neurodegenerative diseases. So, you know, neurons, they pass signals. That's how we sense pain or smell. And they have a very sophisticated networking. So there's a 4-kilodalton protein, which is 39 to 42 amino acid, which is derived from amyloid precursor protein. This contains phenylalanine, valine, and APP 670 and 71 contain asparagine and leucine instead of lysine and methionine. So these mutants can be cloned and expressed in mice to understand the pathogenesis of Alzheimer's diseases. Gene, as I think I mentioned in earlier class also, the gene therapy was first tried in blood cancer, and it was approved by FDA. And there are two types of gene therapy: germ cell based or somatic cell based. And these are the strategies for treating genetic disorders. And this is very interesting, that myostatin is a protein that stops the growth of the muscle, that leads to dystrophic muscles. But there is a protein called as follistatin that can inhibit myostatin, and it can lead to normal muscle growth. So a person who has issue with myostatin protein that is preventing muscle growth, this myostatin action could be blocked by follistatin. So a transgenic vector with follistatin expressing gene could be transduced into embryo to take care of the problem, or into the stem cells. So this is a small chapter, so I finish this chapter. Thanks.